our discussion of protein structure. We're going to talk about the different levels of protein structure. What we've seen so far is what we call the primary structure of a protein. So the primary structure is just the sequence of amino acids. So the polypeptide that we built in the last lecture, or the last lecture segment. So this is the linear sequence of amino acids in a polypeptide, and that's going to be different for a different protein, right? This level of structure is stabilized by peptide bonds. And I'll use that term stabilized a lot when we're talking about what holds together these structures. So stabilized just means what it's held together by. And it's held together by peptide bonds. The individual amino acids are covalently linked. So those are covalent bonds. And if you compare a covalent attraction or a bond to any of the other attractions we've talked about, this is the strongest. So in an aqueous solution, it's stronger than an ionic attraction, it's stronger than a hydrogen bond, certainly stronger than a van der Waals interaction. So these are permanent linkages unless you break them with an enzyme by hydrolysis. So this primary structure then is going to be the most fundamental level of organization. It's going to determine all the other levels. So it's going to determine how the protein will ultimately fold. So here's a folded protein. This is that alcohol dehydrogenase protein again. And what you see in this depiction, in this illustration of the structure, are a lot of individual shapes within the overall folded form. So what's being illustrated here to a certain extent is secondary structure. So the secondary structure refers to those regular repeating structures that are inside the folded polypeptide. So in purple here and green, you see some elements where you see that same shape again and again. So these structures are stabilized, in other words, held together by hydrogen bonds between the peptide bond groups. So we're talking about interactions just between the carbon-nitrogen backbone, not involving the R groups at all at this point. And that's what forms that secondary structure. So these are regular repeated structures. There are two different types of secondary structures. Put this back in here. And one type is a beta-pleated sheet, and I'm showing this first just because this is you can see clearly how the hydrogen bonds are formed between the peptide bond groups. So here's the peptide bond group, here is the carbon and nitrogen, the carbon double bonded to oxygen, and the dashed line there is a hydrogen bond to another part of the polypeptide chain, this time the nitrogen and hydrogen of the next chain, next part of the chain. So this is how these structures are going to be held together. It's going to be hydrogen bonds between the peptide bond groups. So we'll see something called a parallel beta pleated sheet here. We'll also see anti-parallel pleated sheets. You can see this is a little more organized looking because the peptide bond groups line up just so they make regular repeated hydrogen bonds. And so this is going to be fairly stable because there are so many hydrogen bonds in these structures, and they're not just randomly placed throughout the structure, but they're at repeated intervals. The one we're going to talk about first, though, is this one, which is called the alpha helix. Now, in this diagram of the polypeptide, you can see the alpha helix, the polypeptide is shown as a coil, and the only parts that you're seeing of the polypeptide is the backbone itself. So you see the peptide bond group, which is the same thing you were seeing with these beta pleated sheet pictures. So with these structures, we don't really consider which R groups are there, at least not in what's holding these structures together. We're just talking about the backbone itself. So let's talk about this alpha helix first. So the alpha helix structure is defined as a coiled polypeptide stabilized by hydrogen bonds, but specifically by hydrogen bonds between every fourth peptide bond group. So not just any arrangement of hydrogen bonds, but precisely every fourth peptide bond is hydrogen bonded to the next fourth one in the polypeptide. So that means that every single peptide bond group will be hydrogen bonded to another one. Now here's my attempt to draw that and just to show a single hydrogen bond between two peptide bond groups approximately spaced along at least my illustration of a coil here. And what you'll notice is when this coils, the R groups are just kind of sticking out like fuzz on the outside of the coil. So they're not directly involved in the interaction that causes the coil to happen. And what you'll also notice is often when you see these structures, they're shown as a ribbon. So here's another way to depict this alpha helix. So you can see the hydrogen bonds here, spaced every fourth peptide bond group that are holding this in this coiled form, but also the polypeptide itself is shown as a flattened ribbon. So you'll often see, here's another depiction of it again. Like you'll often see this shown as a flattened ribbon. So why does it show it flattened in this way? Well, you may remember that that's because of the peptide bond has some double bond characteristics, right? So it's planar, it's flat. And so since every one of these peptide bonds is planar and flat, there's no rotation around that bond, almost like a double bond, then you would expect the whole structure to be flattened and ribbon-like in that way. And so that's why the alpha helix is often shown as this flattened ribbon, because it truly does flatten out the shape of the polypeptide chain. So these alpha helices can be very short as like 4 amino acids, well, 5 amino acids probably, but that's about as short as they can be, and they're as long as 40 amino acids. And because every single peptide bond group is hydrogen bonded to another, so that's a lot of hydrogen bonds, then that provides a lot of strength to this structure. It's very difficult to disrupt. So, I like to draw things as just a line. Remember I drew the amylose as a coiled structure like this, and you wouldn't be able to tell this from my drawing of amylose because it's just a coil, it's just a helical form, except that I'm labeling this polypeptide backbone. So well, that must be the polypeptide backbone, and those little red lines are my attempts to draw the many hydrogen bonds that's holding this together in a coiled shape. Now there is such a thing as what we call an effect of the R groups, by the way. There's an R group effect that will determine whether or not a certain sequence of amino acids in a polypeptide are more likely to form this helical structure. So the R group effect just refers to the fact that there are certain amino acids that, because of their size or their shape or their charge, won't sit quite right next to each other in a coiled structure, but so they may be less likely to form one. But the R groups themselves, as I said, they're just sticking out to the side here. They're not directly involved in it. But this is a very stable structure because of the hydrogen bonds. You may remember earlier in the semester I mentioned that hydrogen bonds would be very important, and they continue to be. It's what's going to hold these structures together. So here's kind of a ball and stick model sitting on its side that give you an idea of the way the polypeptide chain would have to coil to form this structure. You may have an opportunity to build at least a partial polypeptide chain, depending on how many carbons you have in your organic chemistry model kit, assuming you have one. But you'll get an idea of how it would have to twist in order to have those hydrogen bonds form. So when we see this is the lysozyme structure again, just showing you the amino acids linked together. This just shows you the sequence of amino acids, but what we find is you can kind of tell, and they're kind of color coded there. There's a yellow one and then there's a red one. There are areas, domains, or parts of the protein that are more likely to form an alpha helix because of the amino acids. And so what you usually will see is what we call a ribbon model, or ribbon structure model of a protein. And the amino acids, the alpha helices would be depicted in this way. And so they're just color coding it to distinguish one alpha helix from another. So you have some proteins, like lysozyme is a very small protein, and you can see a lot of its structure is in alpha helices, but there's some other shapes in there, and then there's some of the shapes are just shown, instead of the coiled flattened part, it's just a thinner chain, and that's an area of what we call random coil that has neither an alpha helix or a beta pleated sheet. So I'll get to the beta-Cletus sheet in a minute. So we're going to see mostly proteins like this that will have a combination of different secondary structures within their overall structure, but there are some proteins that have just secondary structure. Now some of them have secondary structures, alpha helices often, that are held together into larger structures. So we would say they have quaternary structure, which means it's more than one polypeptide. But consider the alpha helix here. It has those R groups sticking out on the edges that you can't really tell in this picture. And what happens is often those alpha helices get coiled together, stuck to basically other alpha helices. So you see a structure like this. So this is showing you 3 different alpha helices, just shown as coiled ribbons here. And their R groups are interacting and holding them together into almost like a fiber. And so there are some proteins we call fibrous proteins because they form these long fiber-like structures that are just coiled alpha helices held together. Examples of this that you should be familiar with would be collagen is one. I think this picture right here actually is this illustration is depicting collagen because it's got 3 different alpha helices coiled together to make one fibril. And another one is alpha keratin. Alpha keratin being the protein in hair. So let's consider collagen for a minute. So different ways we can depict this alpha helix. So here's one alpha helix. You can see the ribbon structure superimposed on kind of a ball and stick model there. And then if we show more of a space-filling model, an alpha helix might look something like this. And then in this third picture, you're seeing the 3 different alpha helices all coiled together. So you have a gray one, a light blue one, and a dark, I guess, purple one. So all 3 of those are making one fibril of collagen. And so this is what collagen looks like under a scanning electron microscope. Collagen is the main structural protein in the extracellular matrix, which is the stuff right outside of the cell membranes, in between cells. And this is found mostly in connective tissue. It's the most abundant protein in mammals, like 25% of the protein in mammals. It's what you find in a lot of in things like cartilage and bone tendons and ligaments and skin, but you find it in basically all tissues in that extracellular matrix. So we say it's a structural protein, which means its function is just to be a structure in the organism or a structure in the cell. It's not acting as an enzyme. It's not doing any other function. It's just forming a part. So another structural protein, in addition to collagen, is alpha keratin. As I said, that's the structural protein filament that's in hair. Now, when I looked for that other picture of collagen, I just Googled collagen to see what would come up. Unfortunately, when I Googled alpha keratin, this guy came up. That's quite a lot of keratin there. But I had to use it. I mean, it came up, right? But if we get an idea of what alpha keratin looks like, it's very similar to collagen in that you have this ribbon would depict a single alpha helix, and you have 2 alpha helices coiled together instead of 3. There's 2, and then you have what's called a protofilament when you have your coiled coils, and so you keep coiling together the already coiled 2 alpha helices, and you come up and you form a filament, and that's what hair is made of. These twisted filaments. So they're just alpha helices twisted together. It's just a lot of them twisted together. And they can become untwisted. In this picture, you see more of the ball and stick model showing you the alpha helix, and there's a nice alpha helical coil here. And this part is coming a little bit uncoiled, and then that's an area where you're having an uncoiling, kind of a splitting of the hair filaments, responsible for something like split ends, for instance, kind of coming apart. So those are structural proteins that are made entirely of one type of secondary structure, in this case, the alpha helical secondary structure. As I mentioned before, we're mostly going to see, other than in those structural proteins, we're going to see proteins that are composed of lots of different secondary structures that are all folded together to make what we call the tertiary structure, or the whole protein. This is lysozyme again. This is that spike protein, that surface protein on the COVID-19 virus. We'll be talking a little bit about this you can see it's a very complex protein. We'll talk a little bit about how it works, but one thing you'll notice in this picture, this is the protein, that allows the virus to attach to a cell, to infect a cell. Well, you'll notice in blue here a lot of alpha helical areas. There's also some other areas that are more flattened, looking, like in this area here that's also in blue, those are not alpha helical areas, but they are secondary structures. Those are the beta pleated sheet structures. So you have both types of secondary structures in there all folded together to form this larger structure. And we'll talk about how that larger structure ends up being formed as well. But first, let's take a look at the basis for this other secondary structure, the beta pleated sheet. So we compare these protein secondary structures. What they have in common, of course, is that they both are held together by hydrogen bonds between the peptide bond groups. That's what defines a secondary structure. It's a regular repeated form based on hydrogen bonds between peptide bond groups. But we know that the alpha helix is specifically when you have every fourth peptide bond hydrogen bonded to another. In a beta pleated sheet, it's different. You're going to have these segments of a polypeptide that are hydrogen bonded to each other. So beta pleated sheets are defined as layers or a sheet of these bent polypeptide segments. You can see these are bent like an accordion pleat here. That's what we mean by a bent. And they're held together by hydrogen bonds, but between these segments, and these can be parallel or anti-parallel. So the one that's shown here, this is anti-parallel. One's in one direction and then kind of turns and comes back in the other direction. You could have it parallel when they both run in the same direction. But again, you'll notice that you have this flattened structure and it has this bent kind of accordion pleated structure, which is why it's called a beta pleated sheet. So why is it pleated? What causes that bending? It's the same thing that causes the ribbon shape in the alpha helix. It's that double bond character of the peptide bond. So it's planar, there's no rotation around that bond, and that's what causes these pleats to form. In some polypeptides, again, depending on the R groups that you find in a sequence, it's either going to be more likely to form an alpha helix or possibly more likely to form a beta pleated sheet. So it's due to that planar peptide bond. Everything looks flattened, and that's why often you'll see the alpha helix and the beta pleated sheet is shown in this ribbon-like structure. So why does that planar peptide bond cause the bending? Well, let's take a look at just a little segment of a polypeptide. Now, I've just drawn a little bit of a segment here. So it's alpha carbon, and then you can see here's the beta pleated. It's not a complete polypeptide. I'm just showing you two peptide bonds. Now the dotted line there to remind you that that bond between carbon and nitrogen is somewhat planar, somewhat like a double bond. So the alpha carbons, you recall, are the central carbons where the R groups are attached. You'll notice I've drawn this so that you have the R group pointing up in this case, and in the next one is pointing down. So it's twisted upside down. You can see the peptide bond group is twisted upside down too. And that's because if you consider those alpha carbons, that's just like a normal on either side, the bond on either side of the carbons, the alpha carbons, it's just like a normal single bond. So you have a 180-degree rotation around that alpha carbon. So that means that these can twist in either direction. But if you consider the peptide bond groups, right, those are the planar parts. And so consider a flat plate there. Right? So that's the planar part. The whole peptide bond group is flattened. So this one is flattened too. But because you have this rotation around the alpha carbon, this could twist in either direction as long as both of those sides stay flat, essentially. So this is how you get that accordion shape. You can just, if you remember this strip of paper that we could use to coil into an alpha helix, if you took that strip of paper now, you could pleat it like this. So you just bend the piece of paper into a sheet, and where you're bending at the point of where the fold would be, and yes, you should try this at home, the point where the fold would be, that's the alpha carbon at the fold, right? And then you have the flat part. So here's the first flat part. Here's one peptide bond, and then here's the alpha carbon, and then another flat part is the next peptide bond. You'll notice now the carbon double bonded oxygen is pointing down. And then we have another 80-degree turn at the next alpha carbon, and this one is going to be facing up, and then another alpha carbon in the next peptide bond group is facing down, and we have our pleat in this small section. So this is just one segment, but it's bent this way. So if we have more than one of those segments, so here's the segment I had shown a moment ago. Here's just some random coil area just to put these, I wanted to make these anti-parallel to each other. So here's one segment, here's another segment, and those segments of pleated polypeptide are being held together by hydrogen bonds there in between the peptide bond groups. Remember, you have this peptide bond is going to be pointing, or the carbon-oxygen part of it is going to be pointing down, and the nitrogen and hydrogen could be pointing up in the next one, and they can hydrogen bond together. So this might be, you might find it useful to take a strip of paper again and bend it in this way, draw your peptide bond groups and bend it and see if you can line this up. Now again, we're not talking about, we don't even need to put the R groups on our strip of paper when we draw this carbon-nitrogen backbone, because the R groups are not directly involved in this interaction. It's just between the polar bonds of the peptide bond groups themselves. So where are the R groups there? The R groups are just above and below the plane of those sheets. So you have R groups sticking out here and R groups sticking below, which are not really possible for me to draw very well. Here's one sticking down, these are sticking up. So it's hard to show a three-dimensional structure in 2 dimensions, particularly when I'm drawing it. That's why I didn't do a live drawing of this, because I'm sure I would have messed it up. So we could rely, I think, more easily on pictures from the textbooks, because they can have the whole shading thing, give you that three-dimensional look. Now you can tell this goes up, this goes down, up, down, and you can tell more that it's pleated. So, but this is basically what I was trying to show, and you can see the nitrogen and hydrogen bonded to the carbon and oxygen of another peptide bond group. Now here's one with there's 3 of them, so you don't have to just have 2. Here's one segment going one direction, the second segment going in the other direction, third segment going the other direction. They're not showing that these segments would be, there would be a continuation of the polypeptide here. We're just showing you the pleated segments. And then the hydrogen bonds, the dots there between the polar R groups. So this is one beta pleated sheet. This would be a flat sheet. And so if you think about these ribbons would be next to each other. And their R groups would be pointing up from the sheet. So if this purple is one flat sheet of paper, and it's up against the screen here, then think of the R groups as coming out at you from that flat sheet and going away behind the plane of the board or screen. Let's see if we can attempt a little better view of that. So I tried to find something where you can get an idea of the three-dimensionality of this. So here's one beta pleated sheet, part of it anyway. Just showing part of one. And you can see they're trying to show the R groups pointing up and down. So these beta pleated sheets can stack onto each other. So you have these layered sheets because the R groups are pointing up and down. The R groups now can interact with each other to kind of hold the sheets together. So what you find with the beta pleated sheet structures is they tend to stack like this, but because the only thing that's holding them together are usually these very weak interactions between R groups, they can peel apart pretty well. So you can peel beta pleated sheets apart. So there are some structural proteins that are made this way, just of these layered beta pleated sheets, and one of them is beta keratin. So it's related to alpha keratin in hair, but this is the structure in fingernails and other structures in mammals, horns and things like that, that's called beta keratin because it's beta pleated sheets. And you can see in this picture that the sections of it can peel apart. You're not seeing individual molecules there, so you're not seeing individual layers, but you can see where the layers have kind of peeled apart a little bit. Another example of beta pleated sheet structure, a structural protein, is fibrin, which is the silk protein in spider webs. So this is beta pleated sheet, and that's what lets that's why the structure can stretch and recoil a little bit, because the sheets are only held together by weak interactions between the, not the sheet itself is held by hydrogen bonds, but the stacking, the layering of the sheets is held together by these weak interactions. Here's a ball and stick model to try to show that. So here you can see, here's one beta-pleated sheet. Here's another one. And the R group, so this is the R group from alanine, and alanine is one of those very nonpolar groups. And so this R group sticking down from one beta-pleated sheet and R group sticking up from the other beta-pleated sheet. And alanine is very nonpolar. So the only kind of interactions you're going to have between those sheets is just van der Waals interactions. So that's why this makes a very stretchy type of structural protein. Very easy to pull those sheets apart from each other ever so just a little bit, and then they come back together. So you'll see a lot of these ribbon diagrams to show these secondary structures, the alpha helix, the beta-pleated sheet, and I would expect you to recognize those as secondary structures when you see them. Now you know that's what a flattened coil means in a protein. Now you'll often notice with the beta-pleated sheet they'll have these arrowheads, so it's just showing you the direction of the polypeptide. So it goes from the amino end to the carboxy end and around, and then so this is showing you that it's anti-parallel. So in a structure that has beta-pleated sheets, you might see anti-parallel ones, and the arrows tell you the direction of the polypeptide, or you might have parallel beta-pleated sheets where all of the pleated sheets that are hydrogen bonded together to make that layer, they all face in the same direction, which means there has to be some random coil, some non-beta-pleated area to allow them to go back in the other direction, right? So both of those are found as parts of proteins. So what we're going to see mostly as we start into our talking about proteins like things like enzymes and protein hormones and different types of signaling proteins and receptors, all the different type of protein functions we'll be talking about. The vast majority of them will have more than just secondary structures. They'll have what we call tertiary structures, where we have this folding of the secondary structure superimposed to make a larger, more complex structure. But within that structure, you often have the beta-pleated sheet and the alpha helix, which is what you see in this COVID-19 virus spike protein, right? You see a lot of alpha helices in here, and you see beta-pleated sheets. And we're going to find that those domains that have alpha helices, for instance, tend to have certain properties that allow them to function in a different way. For instance, in the COVID-19 protein, the long alpha helices are involved in the fusion area of the spike protein that allows it to help fuse the virus to the cell receptors as they move. And we'll see that beta-pleated sheets tend to have a kind of an opening and closing mechanism sometimes. So we'll talk about how you get these larger folded structures. In the next lecture, I'm going to talk about protein tertiary structure, which is the next level of protein organization, and we'll see these more complex structures, and we can start to understand how you get these big globular structures. This is this second picture here is a space filling model of an enzyme called trypsin. This is a digestive enzyme, and what you're seeing are the parts of the parts of the polypeptide that are on the outside surface of this larger complex structure. So certain amino acids will be towards the outside, some will be on the inside. How does a long polypeptide chain fold into a very specific shape like this? And that's the type of question we're going to talk about in the next lecture.